Hi, my name is Christian Peterson, and I'm a fifth-year PhD student in Dr. Woo-Suk Chang's lab here in the Department of Biology. I'm here today to walk you through my poster, which covers the comparison of commercial soybean varieties that were planted with our lab's drought-tolerant bradyrhizobium inoculant under irrigated and non-irrigated conditions at the Sandhills USDA Research Station in North Carolina over 3 successive growing seasons. Bradyrhizobium inoculants are a type of plant growth-promoting rhizobacteria that fix atmospheric nitrogen into usable forms by the plant through formation of root nodules. Now, drought is a very big impediment to this by not only killing off the symbiont, but also reducing the amount of photosynthates the soybean plant can deliver to the symbiont. And with rainfall expected to decrease up to 20% by the end of the century, studying this symbiosis is crucial to maintaining sustainable soybean production. The way we studied this was by determining the genetic effect of drought stress on bradyrhizobium, through which we found drought-specific genes that were upregulated, which gave us a molecular marker to test a lot of isolates that we had taken. Through this, we found Bradyrhizobium japonicum TXVA. Sandhills presents some very unique soils. The sandy loam quality of the soil there is essentially like sand planting on the beach. Are very droughty and do not sustain rhizobium very well from season to season. Interesting fact about it is even the rain-fed portion had to be irrigated until the reproductive growth stage, or else the plant stand would have been so poor that there would be no beans at all. So from 2019 to 2021, we maintained irrigated and rain-fed randomized complete block design plots that grew in cultivar each successive year. Starting with the commercial cultivar N7003CN in 2019, the slow-to-wilt or drought-tolerant cultivar Woodruff in 2020, and then the fast-to-wilt or drought-sensitive cultivar NC Roy in 2021. Now, standardized inoculant density and application amounts were maintained throughout each year to make an equal comparison. And then also sterilization of the planter was done after each treatment was planted to prevent any kind of cross-contamination. If you'd like, you can scan the QR code. It's on the poster, and you can see a drone video of the field being planted in 2021. Our mid-harvest metrics show increased growth via dry plant biomass readings and increased nodulation, especially in the rain-fed portion. Looking at final yields, 2019 shows the greatest yield increase of 11.5 in the irrigated and 29.3 in the rain-fed. It was also the year that suffered the most drought as recorded via high temperature and low rainfall. Another interesting result was despite the promiscuous nodulation in the drought-sensitive NC Roy, that was the only cultivar that showed increase in both irrigated and rain-fed portions in 2021, indicating a potential symbiotic effect or additive effect of our drought-tolerant inoculant with the drought-sensitive cultivar. Across all 3 years, plants inoculated with the TXVA strain showed increased final yield in 9 out of 12 cultivar treatments and had a total yield increase of 3.9%, while having a rain-fed yield increase of 10.4%. Future directions for this project are determining an alternate formulation of inoculant carrier that imparts greater desiccation tolerance for our inoculant. Also, we can develop a molecular tag to identify the abundance of TXVA in the nodules to determine if the symbiotic effect is strictly from this organism or a communal effect in the microbiome. Then also determine alternate nutrient solubilization potential of the TXVA inoculant. Thank you very much for listening to my talk.